Interesting presentation, Mr. Adrian Kryk. Our next speaker is Professor Yenny Risjani from University Brawijaya. Professor Yenny Risjani is got her education in Nantes University in France. Her fields of expertise is marine biology, phycology, ecotoxicology, algae bioactivity, and biodiversity. Ladies and gentlemen, please welcome Professor Yenny Risjani.
Thank you very much, Martha, for your introduction. And last webinar, I shared you about the microalgae, about Porphyridium and Haslea. But today, I would like to share with you about macroalgae, Kappaphycus alvarezii. Sorry, it is already. You show the screen?
Yes, but it's not full screen.
Okay, I will share the slideshow. Okay, the title is about Kappaphycus of the brown and green morphotype, and we can see that there are two types of macroalgae in Indonesia: brown and green morphotype. So we use the title pigment or genetic. This is our question because until now the information about what can make we can characterize the microalgae by pigment or genetic is it's very scarce in Indonesia, and this is why we use the title. And for the genus Kappaphycus or Euchema in Indonesia, this is very general in Indonesia. Genus Euchema or Kappaphycus is naturally common in the coastal area of Indonesia. And we can see the local name. We can give the name of Sakor from several area of island, and Tambalang and Kotoni. Sometimes we call this Kotoni, or we call also Sumba in the area of in the island of Sumba. And sometimes we call this type is Euchema or Kappaphycus. Yeah. Sometimes the farmer use Euchema, but the scientists use Kappaphycus for the present time. The genus Euchema firstly published in the original publication by Agathe in 1847. He introduced the genus from Mexico, and in 1930 the name changed to Euchema cottoni by Weber, and it also has other synonym, heterotype synonym, Euchema cottoni, and this is from Weber Bosch And it changed in 1985 by Doty using Echema alvarezii with homotypic synonym Echema alvarezii. And in 1992, because Trono found that in this genus, he found the Kappa, carrageenan type Kappa. So he proposed other name, Kappaphycus alvarezii or Euchema cottoni. Euchema cottoni normally used by, generally used by farmers, by economic purposes. And for the last, until now, the name changed to Kappaphycus alvarezii. Yes, that because the source of Kappa carrageenan type in Euchema cottoni. So Euchema cottoni is a commerce name. And there are two different morphophytes in Indonesia, the red and the brown and the green one. And information based on their biological characteristics such as growth, pigments, and hydrocolloid is important to facilitate algae production and to get benefit from biotechnology. Sometimes it is hard to determine what is the name of the genus because by morphological feature it is sometimes we confuse with the genus or the species name. So it is very useful if we can use other method to for taxonomy. And we can see the taxonomic identification of carrageenophyte red marine algae is confused due to lack of adequate characteristic. This has been written in the published journal. Thank you. This is my ex-student, Gunawan Abidin. And there are also difficulty when analyzing the species relationship between color, morphotype, and site of origin. And the use of molecular tool has been promising in identifying the similarity and genetic gap between the variety from different locations. It is useful in seaweed selection to obtain superior heredity and genetic resources. Deep diversity provides opportunity for breeders to obtain new strains or improve variety with more desirable characteristics. Previous genetic study, Kappaphycus and Euchema, has been published in China and also in Hawaii and Philippines and in Malaysia, but from Indonesia it is very scarce. And until recently, there is no genetic information about the two varieties originating from coastal area. In this case, Lombok, South Sulawesi, and Madura. This is the reason for conducting a molecular study to explore genetic variations, especially in green and brown morphotypes of Kappaphycus alvarezii. Higher resolution of RAPD can be reliable, used for variety level differentiation of algae strains. In fact, it has been used to determine genetic diversity in germ plants, like in kelp or RAPD, also relatively simple, easy to prepare, has faster yield, and does not require any genomic background. And furthermore, it has been proven to be reliable, easy to generate, and inexpensive. And this is the localization, the culture in Madura, in Lombok, and in Sulawesi. And the study aimed to evaluate the relation between age culture on ground, pigments, and its carrageenan content in Kappaphycus alvarezii of two different varieties, brown and green. We also compare the genetic similarity between the green and brown morphotypes of Kappaphycus alvarezii that inhabit different sites in Indonesia. The influence factor affecting color morphotypes in view of genetic and pigment content can be obtained. And we use DNA isolation, which was conducted using the phenylchlorophore method, and DNA amplification using PureTech FTG PCR bed, and RAPD using unweighted pair group method with SEN. And in this RAPD screening, 5 primer, which showed polymorphism, were subsequently used in this study. There are 5 primers: CA1, CA2, P40, P50, and the ALRP. And we can see here the optimization of genome concentration and screening RAPD primers using for use if we use below 50 nanogram or smaller DNA concentration, it will exhibit unclear band of high molecular weight position. This is the 50 different concentration of CA1. So therefore, the use of 50 nanogram concentration and above of DNA produce a relatively clearer fragment than others. And the largest range of genome concentration show the position of more amplified fragment ranging from 250 to more than 1,000. And the primary primers with GC proportion of 25% and 47% were used in the study. And for the grouping of the species between green and brown variety, this is the amplification using P40 primers. And this is the row show the fragment between species or specific marker. And this is the clustering, UPGMA cluster of seaweed from the green and brown variety from South Sulawesi. And we can see the species of green from Sulawesi has a higher genetic variation, and but for the brown has lower variation than the green one. And for this one, this is the green morphotype. with UPGMA cluster for green with different different site location. This is the same group with the same morphotype, but different location. And we can see here there are high difference variety of the different seaweed cultured in the different location. And for brown morphotype, also this it gives this for example Pinrang and Pinrang or Sulawesi this form the same group, but it is it show high difference with Madura and Lombok. A wide range of phenotype can be produced when the same genotype is subjected to different environment. And for the genetic gap of brown Kappaphycus, alvarezii from Lombok, South Sulawesi, and Madura, so that the genetic gap of brown morphotype from the three sites is large. And the analysis of Kappaphycus alvarezii genetic relationship from 3 different sites using 5 primers showed a high gap and variation. The green and the brown morphotype have a closer similarity than their cultivation locations. What is the advantage? In fact, the gap and proximity between the samples determine the success of crossbreeding and cultivation. And this is the advantage for the study. Right? A wide range of phenotype can be produced when the same genotype is subjected to different environments. However, the relationship between genetic gap and hybrid performance is variable, and it depends on environmental factors and material used. The more diverse the genetic, the higher the chance of becoming superior genotypes. This means variability has breeding advantage to obtain superior heredity. Furthermore, diversity and variety in genetic resources provide possibility for seaweed breeders to develop new strains and improve species with desirable characteristics. And we observe various studies using the same genus Kappaphycus and or Akema. They are from Hawaii with using partial nuclear RNA and also and DNA barcoding like approach, and the other is have been done in Philippines using DNA marker CO15P, and this is the mitochondrial DNA, and from Southeast Asia, this is also with KOX1 and KOX2 spatial genetic marker, and from Malaysia with mitochondrial COX1, COX2, and other. And for the study, we use three locations, and for sign, it has been used with ITS sequence. Suggestion for this after this study, based on experience from the research with RAPD being successful in molecular identifications require certain conditions. First, the use of primer with the right GC proportions. The primary primers with GC proportion, certain proportion will give the best result. Second, the use of a higher DNA concentration. So why? That because smaller concentration exhibit unclear bands of higher molecular weight position. Meanwhile, larger concentration of obscure bands tend to be obtained at low molecular weight position. Also, the use of 50 nanogram DNA produce a relatively clearer DNA fragment. Now, for this finding, for the both morphotype, brown and green, there are smaller genetic gap between the morphotype, but for the location, there are higher genetic gaps between the different location. So green and brown Kappaphycus have closer similarity than their location of origin. And for breeding objective, it is suggested to select Kappaphycus alvarezii based on variability of location. And color of thallus is depend on the genetic factor or the pigment content. So other related study are necessary to understand whether both morphotypes are dependent on their chemical characteristic or not. We have data with the growth rate and the growth, seaweed growth with both morphotype. For the young, the brown eczema grow faster than the green, but it is changed after 15 days. The green has higher specific growth rate than brown eczema. So the older stage for more than 6 weeks, it reached the same weight. And we have also data about the chemical content, ocaraginan, chlorophyll A, phycocyanin, and phycoerythrin in the species. And we can see at first the young brown grown faster, but then both morphotypes reach the same weight after 4 weeks. And for this one, we can see chlorophyll A increase positively in line with thallus age. Brown strain has the same chlorophyll A as brown strain, but it's only a little difference. But with the statistical evaluation, we can see this is the same. And for phycoerythrin, the brown strain contain more phycoerythrin than green strain, and phycoerythrin increase positively in line with thallus, and phycocyanin increase positively in function of thallus age, and brown strain contain more phycocyanin than green strain. What is the most influencing in characterization between the morphotypes, variety, pigment, or genetic? So our research show that variation between color morphotype did not represent higher genetic gap. Yeah. But although genetic factors play a role in pigment biosynthetic pathway process in the cell. This is the biosynthetic pathway process. It is started by nuclear gene transcription in the nucleus, and then protein import and processing in the membrane cellular membrane, and then there is a process of gene transcription and translation, and protein folding and degradation here. And finally, thylakoid formation and pigment biosynthesis is formed in this chloroplast. and then it entered to chloroplast division. This is the typical pigment in red algae. So the design of the picobilisome is such that excitation energy is delivered from any one of these many chromophores to reaction center in the photosynthetic membrane with an efficiency approaching 100%. And for this, this is the stroma and the thylakoid membrane here, and this is the lumen. And for picoerythrin and picocyanin play a role here in the stroma, and for the other pigment, it play here in the membrane. For example, for chlorophyll A. And for picoerythrin and picocyanin are more represented in the brown morphotype than in the green. So this is the light energy transfer pathway in algae from picoerythrin to picocyanin, and then to allopicocyanin, and then to chlorophyll A. So this is the outdoor culture in coastal area with more chlorophyll A content was relatively the same between the brown and the green. We can see for the green one, they have this relatively the same chlorophyll A content, but it is different with picocyanin and picoerythrin. Conclusion: the plant age was very significantly has a positive correlation on all pigments. In this case, chlorophyll A, picoerythrin, and picocyanin pigment. All chemical contents, including carrageenan, increase as thallus egg increase. Brown and green strain of Kappaphycus alvarezii has a short genetic distance. That is mean they have genetic similarity, but the brown morphotypes are characterized by the higher value of picobilin pigment than the green. The brown variety receive energy to form picobilin protein in their stroma, then transfer it to form chlorophyll A in the thylakoid cellular membrane. This is my presentation. Thank you.
Thank you so much for your very interesting presentation, Professor Yenny Risjani. I would like to remind that you have the opportunity to submit the text question to today's presenters by typing your question into the chat box. You may send your question at any time during the presentation.